Hello, everybody. My name is Orlin Todorov, and I'm a PhD student at the University of Queensland, and I'm about to present you some of my work on the evolution of the mammalian brain, where I focus on 3 different mammalian clades. So the mammalian brain is an extremely diverse organ. Its weight ranges from under 1 gram in certain shrew species up to more than 8 kilos in the sperm whale. So some of these brains share larger portions of their evolutionary history than others. And a way to account for that are the so-called phylogenetic comparative methods, which account for the fact that certain species are more closely related than others. And so we should treat them in a different way statistically. So if we are about to ask questions related to brain evolution, there are mostly 2 different types of hypotheses. Some of them are selection-based, and they mostly explain what in the environment selects for larger or smaller brains, and such selective pressures might be social or individual. So one of these hypotheses is the social brain hypothesis, another hypothesis is the ecological brain hypothesis. Alternatively, certain hypotheses about brain evolution revolve around constraints around brain variation, and they try to explain what constraints brain size increase. At the same time, it is still unclear whether we should treat brain evolution as a mosaic process, so whether certain parts of the brain evolve independently from each other, or whether the whole brain is under certain developmental constraints, so we should look at the whole brain, or whether we should look at different functional areas or different cytoarchitectural areas. Another issue that is not very clear in the field is whether we should ask questions related to brain evolution on whole phylogenetic groups like phylums. So whether we should look at the whole vertebrate phylum, or whether we should look at smaller clades like mammals or reptiles, or even further, maybe we should direct our inquiry towards smaller infraclasses like marsupials or placentals. Or we can even go further down the phylogeny, like we can look at different orders like primates or rodents, or even suborders or families. So what I did as part of my PhD thesis is I looked at 3 different taxonomic groups, which span different number of species and have different variability. So one of them is the primate order of around 300 species. The second one is the marsupial infraclass, which also coincidentally has around 300 species. And lastly, I looked at leporids, which is a small family within the lagomorphs with around 75 different species. I also looked at different selection and constraint-based hypotheses, and I also looked at the question whether the brain evolves in mosaic fashion or more in a concerted unitary fashion. So in my first study, we looked at the primate hippocampus. That's quite a complex part of the mammalian brain, which looks like a seahorse in certain species and not that much like seahorse in other species. Its function is related to learning, memory, spatial social information, emotions, and many other things. So I tried to look into whether hippocampus size and organization is related to several different traits like home range, diet, or group size. And for group size, I used 4 different measures derived by 4 different research groups. So it's interesting to see whether they agree or whether they reach to the same conclusions. My sample spanned 43 different species of primates. So what we found is that the cornus ammonis complex, that's that part of the hippocampus, it all evolved in concert with the group size. So this is in concert with the social brain hypothesis, while only the CA1 segment and the subiculum could be predicted by home range size, which is more closely related to those individual pressures that I mentioned, or the ecological brain hypothesis. Surprisingly, we didn't find any support for diet affecting the structure and organization of the hippocampus in primates, which is quite surprising because previous research had shown that the primate brain size is predicted by diet, but not sociality. So obviously there are different pressures at play under the level of the whole brain or partitions of the brain. More surprisingly, when using 4 different measures of group sizes, some of them were reaching to the same conclusions, but some of them weren't. So for example, looking at the CA1, we could find that CA1 correlates with the group size with those 2 data sets, but CA2 was correlating with group size with these 3 different data sets, while CA3 was only related to that first group size. So this is a cautionary note of what kind of measures we're using, and being cautious with the data that we are using for our analysis. We also did an analysis called ancestral state estimation, where we can reconstruct some of the evolutionary history of the traits of interest. So on this graph, we can see the total hippocampus volume on one side and the relative hippocampus volume on the other side relative to the rest of the brain or the neocortex. So what we found was that species that have larger hippocampal volumes, that's the blue lines here, they usually have smaller relative hippocampi. This is because the slope of increase of the hippocampus is shallower than the slope of the neocortical increase, meaning that species that have larger hippocampi usually have larger neocortices. So probably neocorticalization or increased size of the neocortex goes hand in hand with increased demand for hippocampal processing. In my next study, I focused on an infraclass of mammals, marsupials. They are mostly found in Australia, but they're also found in the Americas and New Guinea. I used the largest data set of marsupials, 176 species out of around 300. I compiled 18 different traits spanning from diet, activity cycle, gestation length, up to proxies of cognition like play. One big advantage of my study is that I didn't have any missing values, not because I made sure to collect all of them, because most of them are non-existent, but because I used a technique called multiple imputations, where we can impute the values based on the existing values in the dataset. Additionally, I applied the Bayesian generalized linear mixed model approach, which allows for analysis of non-Gaussian distributed data. I proposed a pipeline for conducting phylogenetic comparative studies, where one starting with a dataset with missing values, and all of our datasets have missing values, especially the larger they get, the more the missing values are. Then after analysis of the missingness, so whether the missingness is at random or not at random, whether there's a phylogenetic signal in the missingness pattern, we can impute those values with an algorithm called multiple imputation with chained equations. And on top of that, we also use the phylogenetic version of this algorithm. So what multiple imputations do is that they're not trying to predict the values, so they are not realistic, but what they do is they conserve the properties of the data set, like standard error, standard deviation, means, etc. So after that, I applied a generalized linear mixed model approach and pulled the results from all the imputed data sets. I had 25 imputed data sets in general. The results of this study showed that the only significant predictor of brain size variation in marsupials is litter size. We didn't find support for the social brain hypothesis, but we found indication for maternal investment constraint. So again, applying similar methods in different taxonomic group yields 2 different results. Another type of analysis that we did was trying to disentangle the separate evolution of brain size from body size, because we know that brain size increases in concert with body size. So this method here, called surface, allows us to detect convergent regimes, whether certain species, for example, the red group, we can see here, they've increased both body size and brain size, while other groups, like the purple ones, they have an decrease in brain size, but increase in body size. So this approach is very useful in identifying convergent groups, where subsequently they can be analyzed for similar, let's say, ecological circumstances or similar life history traits. This approach benefits tremendously from inclusion of fossils. So this is something very much needed in the field of brain evolution, where we can calibrate such algorithms with the inclusion of fossils. In the last part of this presentation, I'm going to mention a few words about a study that I did on a smaller phylogenetic group, the leporids. So they're a very homogeneous family. They all have very similar shapes and very similar life histories. I collated a data set of 18 species obtained from endocasts, and this comprises around 25% of all the species in the family. I collected 8 different traits, among which seasonality, litter size, home range, so I could test most of the brain evolution hypotheses. I was mostly focusing on testing the effect of seasonality, and I was able to measure both the olfactory bulb and the rest of the brain from the endocasts that I recovered. The only correlative brain size in leporids seemed to be burrowing behavior and home range. So again, no confirmation of the social brain hypothesis, but this time also no evidence for maternal investment constraint. It might be that leporids are buffering seasonal variation through burrowing and extension of their home ranges, and that's why we don't see any relationship between litter sizes or social group sizes with brain size or olfactory bulb. An interesting observation in this study was that in some of the models, I could see that the lambda estimations, or the estimations of phylogenetic signal, were negative or larger than 1. This is by definition impossible because lambda can only span between 0, meaning lack of phylogenetic signal, or 1 indication of a strong Brownian motion. So such lambda estimations are artifacts of using small sample sizes. They're unrealistic, and they are a result of faulty likelihood estimations. So in such cases, one should just disregard interpretation of phylogenetic signal, which is somehow done, which is sometimes done in the literature, and it should not be done. So in short, I just showed you that looking at different brain partitions reveals a different picture, a different evolutionary picture compared to looking at the whole brain. So both approaches have their utility, and one should tailor their inquiry based on the specific questions that they have. Also, it seems that different selection pressures and different constraints are at play in different taxonomic groups. We could see that we can find support for the social brain hypothesis in primates, but we couldn't find any support for it in both leporids and marsupials. Additionally, data imputations are a very useful statistical technique, which we can use in order not to sacrifice sample size. And there are also ways to correct for phylogenetic non-independence while imputing data sets. Also, one should use caution when using small sample sizes and always inspect for statistical artifacts that can arise from that. Additionally, fossils can allow for further analysis and deeper insights, and they are invaluable for us to be able to calibrate models looking at convergence or ancestor state estimations, etc. At the end, I want to thank my supervisors and collaborators, Alexandra de Sousa, Simone Blomberg, and Vera Weisbecker. And no, these are not their photos. Thank you for your attention.